Can you send a link to the repo? Okay, cool. So basically what I'm going to try to go through is hopefully 3 out of the 4 of these main topics here. The first thing I'm going to do is talk about sort of basic manipulation of data in Python, neuroimaging data in Python, and that's because it occurs to me that some of you just haven't used the Python neuroimaging stack yet. So that should be just a very quick kind of intro. Then I'm going to talk a little bit about the sort of fundamental thing that needs to happen in order to connect this neuroimaging data structure with the tools that we have in the Scikit-Learn stack. And then finally, I'm going to go through 2 applications that you might use in order to ask these questions in neuroscience. Okay. So again, you should sort of use these as you should view these as reference notebooks and sort of inspiration. And I think over the next couple of days, as we continue to work on our projects and stuff like that, you should feel free to ask me or any of the instructors or anyone here who's proficient in Python for a little bit more explanation or a little bit help or whatever. Okay. So the first thing that I want to do is talk about 2 really big packages in the world of neuroimaging. The first one is called NiBabble, and this is in some ways like the centerpiece of the sort of neuroimaging stack in Python. The idea about NiBabble is that neuroimaging data can come in many different shapes and sizes. Some of it's volume data, some of it's surface data, some of it's like diffusion information, some of it's statistical maps. But what we want is one package that can sort of handle all of these things relatively flexibly, and that's what NiBabble is designed to do. The second thing that we're going to talk about is a package called nilearn, and this is effectively a set of tools that allow you to connect these neuroimaging structures with the Scikit-Learn stack and then go back again into the world of neuroimaging. So as Jake mentioned, in Scikit-Learn, you have a number of different functions that can be used to download different machine learning data sets. And as he mentioned, in terms of API, one of the really useful things that Scikit-Learn has done is in some ways kind of define standards for how packages are constructed. And so you'll notice that in nilearn, which is a totally separate package from Scikit-Learn, they've sort of adopted the same structure of having a datasets module and then having a number of functions that begin with fetch underscore. And then you can grab different, in this case, neuroimaging data sets that way. So that's what we're going to use to go through some of these analyses here. I'm not going to go through all of them, but if you just hit tab after you type fetch underscore, it should just give you a list of the stuff that's there. So to start off with, we're going to play around with this Atlas dataset, the MSDL Atlas dataset, and just sort of explore a little bit what's inside. The first thing that I should note is that almost all of these datasets have some kind of metadata that comes along with them. So it's not just raw data values, as everybody should always be doing when they package their data, include some information about what that dataset actually is, so that when other people download it or use it on their own, they can sort of get their bearings that way. So I'm just giving you an example of what that looks like here. This is just a short description that kind of gives you an idea of what's going on here. And as we'll see, effectively what nilearn did was it went up into the internet, it found the URL for that data set, and it pulled it down as this .nii file right here. It's also called a Nifty file. I'm not going to go into too much detail there because I think most people know what Nifty files are here. But we can use NiBabble to very easily load this Nifty file into memory. And to do that, you just use nb.load like this right here. So when we do that, it returns to us what is called a Nifty image object. And this is effectively a wrapper around the raw data itself that gives us some extra metadata and information about it as well. So for example, there is usually an affine attribute, which tells you about the affine transform that applies to that data set, so that you can do things like co-register it with other data. There are lots of other attributes in there as well. There's one that's useful in particular is called the header. And this is another object that has like a lot of other information inside of it. So I'm not going to go through all of that because this is sort of heterogeneous. It depends a lot on the object that you've read in. But when you read in this data, I recommend that you sort of explore through those things to try and get a feel for what you're actually dealing with. If you want to actually gain access to the raw data itself, you can read it in as a NumPy array using the get_data method right here. So if you call this it's just going to return a matrix that represents, in this case, the map corresponding to this atlas. So what we're going to do is just get an idea for what kind of a shape this has. So neuroimaging data usually comes in two different possible shapes, 3 dimensions or 4 dimensions. Those correspond to XYZ or IJK, and some 4th dimension. Sometimes that 4th dimension is time. Sometimes that 4th dimension is trials or conditions or whatever. But that's a common pattern that you see, and something that's sort of assumed in the NIBABEL neuroimaging stack. The first 3 dimensions are always space, and the last dimension is some other thing that you may want to index into. So nilearn provides quite a lot of plotting functionality. It's not just for doing machine learning, but it's also just a set of tools to help you perform your analyses more effectively in general. And one of the most useful things that they have is a function called plot stat map. And what this does is it indexes into a Nifty file and just visualizes that file on a brain. So here we'll just call plot stat map onto the the 4th image that's in this Nifty stack that we've got here. To do that, we're using this function here, index image. And this is a way to tell nilearn, I want you to go into this Nifty file and pull out the nth image for visualization. So we can run that, and we get a nice visualization of that. In this case, each item in the stack corresponds to a different location that has been fit with this particular atlas. So I think this is like a functional covariance analysis that they did to try to define different subnetworks within the brain. So here we can see the 4th network that atlas defines here. And this viewer is a way that you can kind of look at the same data from different kinds of angles very, very quickly. And I'll show you how to do that in a second. So we could easily look at some of the other parts of this atlas if we just change this index right here. So now I'm going to look at the 6th region. I can look at the 10th region. And that's a way that you can very quickly, interactively scan through the data that you've got here. You can perform different operations on the plotting itself. So something that's very common if you're doing, for example, a statistical map like a T-map is to threshold your image. For that, you can do things like supplying a threshold parameter here, and this will only plot things that lie above whatever the threshold that you give. And you can also change the sorts of views that are available when you make the plot. So if you want to display only a particular orientation of the brain, you can do it like this. If you want to display more slices of the brain, you can do it like this. So these are just a couple of examples. Look through the plotting function there and sort of try to get a feel for how you could create the plot that you may want to show in a paper or something like that. And then finally, you can also loop through all of the images in a Nifty stack if it has that 4th dimension. And the way to do that is with this iter_image function. So if you loop through this in a for loop, then on each iteration of the loop, it's going to return the image corresponding to each index that's inside that Nifty file. So here I'm just going to iterate through the first 5 images that's inside this file name so that it doesn't take forever. And each time I'm just going to plot that image. And when I do that, hopefully this isn't going to take too long, you should get a visualization of the first 5 regions that this atlas is defining here. So that's a nice way that you can just quickly kind of parse through the data in order to get what you want out of it. Okay. So that was like very, very quick sort of NIBABEL 101, maybe a bit of like nilearn and visualization 101. This is really just scratching the surface of the API that is available to you here. And what, yeah? What's that?
It plots across.
Uh-huh.
So that's not on where it should be.
On the coordinate.
Oh, okay. Yeah, so there are different, in terms of defining where the cut coordinate is, you can tell nilearn to do that in different ways. So there's I think, a parameter that says like choose the cut coordinate so that it's at the maximum of the activation in the map or something like that. But those are other things that are exposed to you as parameters you can play around with. Also, I'm not going to speak for the data itself because I hold no responsibility for that. So if I show something that's totally wrong, then it's not my fault. Okay. So the next thing that I want to quickly talk about is, all right, so we've figured out how to read in this neuroimaging data into Python. We have it in memory as this nifty object. But how do we actually connect that with something like Scikit-Learn so that we can do some cool machine learning with that data? And to talk about that, I want to cover a really important topic just in general in the field of machine learning, and that's something that's often referred to as vectorizing. So I mentioned before that neuroimaging data comes generally in the shape of IJK and time, or IJK and samples. But that Scikit-Learn almost always expects data to come in the form of a matrix of shape number of samples by number of features. It's going to expect a 2D matrix. And in a simple sense, vectorizing just means taking data of some kind of a shape and turning it into a form of samples by features. And once it's in that form, now it's sort of easily able to be piped into the Scikit-Learn stack. So the trick is that we want to do that in a way that doesn't sort of obliterate our knowledge of what all of these features actually are. And if we take this nice four-dimensional array and we unwind it, so now that it's samples by features, it can be really hard to keep track of like what each of those different features actually maps onto. So the easiest way that we handle this using nilearn is with a collection of classes that try to sort of do some of this magically for you under the hood so that you don't have to think about it as much. So to motivate this, we're going to use a new data set. This is the Haxby decoding data set, and I'll go into it a little bit more detail in the next notebook. But effectively, it was a task in which subjects were looking at, I believe it was images of different kinds of objects. It was like cats and faces and houses and stuff like that. And what they were trying to do is determine if they could decode the kind of object that a person was looking at just using the voxel activity in the brain. And this is going to be the decoding example that we go through in a little bit more detail next. So here we're just going to fetch the Haxby dataset right here. We can see that it's downloaded this Nifty file and saved it onto the disk. So effectively, when we're doing any kind of machine learning in neuroimaging, we're usually doing two things. First, we're pre-processing the data, some amount. Next, we're doing some kind of feature extraction. And then I guess a final third step is after we define the features that we want, we vectorize them so that they're in a form that we can put into Scikit-Learn. So the way that we can do this in nilearn and in the neuroimaging stack in Python is by using some sort of volumetric mask on our data. So usually you say, well, this is the entire volume that I've got, but really I only care about like this subset of voxels that are over here. That you may have determined that experimentally or based off of whatever question it is that you were hoping to ask. So here, this Haxby data set actually comes with a mask itself. And what I'm going to do is use the plot ROI function to visualize what that mask looks like. So again, this is just a binary matrix. It tells us which of the voxels we want to use, or actually, specifically it tells us which of the voxels the Haxby group used in generating the figures for their decoding experiment. And the way that we can combine this mask with our actual data is by using something called a Nifty Masker. So this exists in nilearn.input_data. And Nifty Masker is an object that can be fit to a mask. So that you can create with a mask. And what that does is give you back an object that you can run on a neuroimaging volume, and it will basically return a subset of the voxels in that volume, corresponding to the mask that you want to apply. So here we're going to create a Nifty Masker object. We're going to give it the file name for the mask image that I just plotted above. And then there are a few other parameters that let you do things like standardize the voxel activity across time, stuff like that. And these are things that you may or may not want to do depending on what kind of machine learning technique you're hoping to apply. So when I run this code, what I get back is a masker object that says, this is an object that I can use in order to mask different kinds of things, as long as they have the same shape as the mask itself. So I don't know that Jake went into too much detail on this, but a masker object is an example of what we would call a transformer in Scikit-Learn. This is effectively an object that takes data and turns it into some other kind of representation of that data. That's sort of the most broad general description of that. In this case, it's a transformer that takes volumetric data in 3 or 4 dimensions and it turns it into samples by features. So the way that we can do that is by calling the fit method of this masker object on the, and we can give it either the Nifty object or a path to the file of the Nifty object of our actual volumetric data. And when I call that, it effectively sort of makes sure that the two of these things line up properly. And when I then call the transform function, it's actually going through and manipulating that 4-dimensional array so that it extracts only the voxels that we want, and then ultimately is going to return a subset of those voxels. So the output looks just like regular data here. This is just voxel activity over time. But if we look at the shape of that data, we can see now that it is only 2-dimensional. So it's now in the shape of samples by features. And importantly, this is the shape that we need in order to get it to play nicely with Scikit-Learn. So we're going to go into the actual model fitting and stuff in the next notebook. But the one thing that I wanted to note that's particularly useful for these sort of Nifty Masker style objects is that they can also move in the opposite direction as well. So once we've gone from a 4D array into a 2D samples by features array, we often then want to move back into 4D space. Here I'm just going to do it on the same data, but usually you would do this on, for example, like a statistical map that you would fit using some sort of Scikit-Learn object. And to do that, you use this inverse underscore transform function. So if you remember, fMRI masked is a 2D array. If I use the inverse transform method with that array, then the output is a Nifty image. So it's turned my 2D array back into a Nifty image.
So it's basically doing the masking operation on the array, not on the image, on the vectorizing.
The inverse thing?
No, the masking, sorry. I took a little bit to get started. So the masking operation that it did, it did it on the vectorized portion of the image, not like the whole.
Yeah, I'm not sure how. It may try to be clever with like being efficient with memory, but my guess is that what it's doing is just pulling out data from the Nifty data and copying it and giving you back a 2D array. So it's not changing any of the data inside the Nifty object. Yeah.
It probably also is what it's in the reverse, just through the masks. Where there's one, then just copy it. Yeah.
And so inside of that Nifty Masker object is a bunch of metadata about like the shape of the original data and probably a bit of information that was inside the header, stuff like that. And so using this, we can just return back the results as a Nifty object itself. And then we can do things like plot statistical maps of that Nifty object. We can also like save it to disk, which I'll show in the next notebook. The other thing is that I just want to sort of highlight the fact that the input to inverse transform doesn't have to be the exact same shape as the original data. You can take, for example, like a subset of samples. You could also just take a single sample. If you do a subset, it's going to give you back an X by Y by Z by N samples array. If you do it with a single sample, then you just get back a 3D array, so maybe that's just like a single statistical map. But the one constraint that it has is that whatever you give it has to have the same number of features. It has to have the same number of voxels that are in that mask. They're in the cortical mask. Okay, so this is the way that we can go from a set of neuroimaging 4D data. We can turn it into a Scikit-Learn style n samples by n features array. And now we've sort of gone back to the original 4D data. So what I want to do in the next notebook is talk about that process in the context of a specific experiment in neuroscience. And this is the 1.3 Decoding Haxby notebook for those of you who don't have broken Jupyter hubs right now. Okay, so as I mentioned before, this is data set that the Haxby group has made available to the public, and the developers for nilearn went online and sort of figured out a way to manipulate it and get into a form that you can download as a nilearn data set. This is people viewing images, and the goal is just, can we detect which image people were viewing based off of the voxel activity that was recorded in the brain. So as Jake mentioned earlier, what we need to do is we need to guess whether or not, or we need to predict whether or not a given session was an image of class A or class B or class C corresponding to dogs or faces or cats. And that means that this is a classification problem. Another thing to note is that what we're doing is we need to feed neural activity into some sort of a model, and the output of that model is a prediction about something about the world. In this case, the prediction is what was the class of object that the person was looking at. As such, this would often be called a decoding model. Some people call it a backward model for reasons that I don't entirely understand. But the idea is that you're sort of going from the state of the brain to the state of the world. And that's what we're going to go through in this notebook here. Oh, that's right. So I do want to make a really quick aside and talk very briefly about the difference between encoding and decoding models. I'm not going to go into too much detail except to say that this is sort of like a field of active debate, and I don't want to make any too strong statements because I'll probably anger some of the instructors that are in the crowd. There are people that have lots of different opinions out there. But what I did is added a few links to some useful papers that have come out in the last couple of years that have sort of explored this topic about the difference between an encoding model and a decoding model for the purposes of understanding things about the brain. Decoding models go from brain to world. They, in some sense, flow, quote unquote, against the passage of time, because usually the brain activity is happening in response to things that are occurring in the world. Encoding models go from world to brain, and they more directly kind of ask the question whether we can model patterns of brain activity using these features in the world. You can ask different kinds of questions with these two approaches, and it actually can become a little bit convoluted and complex exactly what subset of questions you can ask with one or the other. So that's why I'm going to sort of punt that question right now, suggest that you guys take a look at those papers, and if you want to have a debate about this, then we can do it tonight after I've had 3 or 4 beers, I would say. Okay, so in this data set, though, we're going to be doing decoding. And we're going to start off the same way as we just did before by loading the Haxby data set as well as the mask itself. So again, here's the mask that we're going to be using. We'll use this subset of voxels to try and decode the category of object that the person was looking at. We're going to feed it into this Nifty Masker, same way as we did before, and then transform the volumetric data, same as we did in the previous notebook. So now we have an output here that is of shape n samples by n features. Right there. So effectively, this is half of the equation, right? In order to do decoding, we both need information about the brain, but we also need information about the world. And that also has to be in a shape that Scikit-Learn can sort of understand and play around with. So included with the Haxby dataset is a collection of metadata that says what kind of picture was presented for each one of these trials. We're going to use a library called Pandas to read that out. It's basically just in a text file with spaces that delimit the different characters that are there. I'm not going to go into a ton of detail about how Pandas works. That's another conversation for a couple of beers later on. Pandas is both lovely and horrific at the same time. It just has a very steep learning curve, but it's amazing and I love it. And I recommend that you all learn it because it's really, really, really useful. So when we read in this data set, we can see that each row is the presentation of some sort of a trial. In this case, these are just REST trials, so there's not anything that's being showed to the person. But if we look at the number of rows that we have in our metadata about the trials, we see that we have the same number of samples that are in our fMRI data set. So probably these sort of have a one-to-one mapping onto each other here. If we print the first 50 values of the labels column, we can see that these correspond to the different kinds of images that were presented to the person. So here they saw a pair of scissors, here they saw a face, here they saw a cat, here they saw a shoe. So to keep things simple here, we're going to restrict our analyses to two categories, faces and cats. I looked, and unfortunately there are no squirrel images in the data set. I know. So the way that we can do that, I've shown you two different ways to do this here. Pandas has a really nice method called eval that basically lets you run different kind of queries, and then it'll return to you a Boolean mask that tells you which rows match that query. You could also use something like NumPy to use logical Boolean indexing to pull out the rows that you wanted to. So here I'm just going to say, tell me all of the rows where the label is either a face or a cat. And what that will return is just a Boolean array that subsets the rows that I want for this decoder. So what I'll do is index into our masked fMRI data. So this is of shape samples by features, as well as into our labels data here. So this is just of shape samples. It's a one-dimensional array. And now I have two objects. One of them is samples by features corresponding to voxels, and another object corresponding to just samples, and that is the category of each trial. This is the X and the Y matrix that we will use in our machine learning model. So what we can do is use really any of the different machine learning tools that are available to us in Scikit-Learn, and it'll pretty much just work out of the box at this point. Scikit-Learn doesn't make any assumptions about what sort of modality your data came from. It could come from a brain, or it could come from a sensor you've got in the ground, or it could come from space. As long as it's of shape samples by features, it doesn't really care. So what we'll do is create a support vector classifier with a linear kernel. This is both like a relatively vanilla classifier, but it's also very powerful and can do lots of cool stuff. And is relatively simple to fit and maybe to interpret as well. We'll initialize our object here, and then fitting this data set is as simple as calling the fit method on our masked fMRI data and on the targets that we're going to have as output. So now that we have a fit model, we can do things like try and investigate the sort of structure that it found in the data itself, and we'll go into that in a bit more detail later on. But as Jake mentioned, a common pattern that you see in Scikit-Learn is that any attributes that are created as a result of fitting your model, so for example, the coefficients of the model, end in an underscore here. So when we fit this support vector classifier, we now create a new attribute called coef, and this is going to be 1 per voxel, and it in some sense reflects the extent to which those voxels are correlated with one class versus another class. The next thing that we want to do now that we have a fit model is that we want to actually validate that model. We need to be able to say, is this a good model or not? Or in a sense, is this a model worth sort of inspecting further and trying to learn something from it? The way that we do that with Scikit-Learn is using this predict method right here. So if we give it a new sort of X matrix, it's going to generate a new set of predictions for the category of image that we've seen. What I'll do here, and this is just for a demonstration, is feed back in the exact same X matrix that I used to fit the model. You should never do this. This is a stupid idea, but I'm just showing this to you so that you can get an idea for how the API looks. And we'll look at things like cross-validation in a second. Scikit-Learn has a number of different functions that can define metrics of accuracy or quality for you. And most of those exist in the scikit-learn.metrics module. So if you do from scikit-learn.metrics import and then you hit tab, you'll see that there are a bunch of different kinds of scores for classifiers and regressors that you can try out on your data. So here we're just going to use the accuracy score function, which I think it just tells us the percentage correct scores, something like this, of correctly identified labels versus the total number of labels that are there. And if we run this on our data, we see that we're scoring at 100%. But that's because we've violated one of the norms of machine learning and tested on the same data that we've trained this model on. So what we really should have done is we should have held out a subset of data, trained the model on that subset of data, and then tested the model on a different subset of data. And the simplest way to do that here is by literally just cutting the data in half. So I'm going to take the first 50% of the data, fit our linear classifier on that, and then I'm going to run prediction on the last 50% of our data here. And when I do that, I can see that we get something that's a little bit more reasonable in terms of the classifier's performance. So one thing that you may have noticed is that I just, in some sense, did my machine learning with a little bit more rigor because I've now separated out into a training and a testing data set. But I've also taken a hit because now I have half as much data that I'm using to actually train this model. So there are a number of different techniques that are out there for being a little bit more intelligent about how you can slice and dice your data so that you're both validating your model in a reasonable way, but you're not throwing away a bunch of your data just so that you can validate that model. This is another one of those topics that is like a beers later on kind of topic because everyone disagrees about the exact right way that you should do this kind of thing. There's another notebook that's packaged with these notebooks that goes through a few different ways that you could do something like cross-validation. And I'll show you an example of this in the encoding model notebook if we have time to get to it. But suffice it to say, there are a lot of techniques for doing cross-validation. Almost all of those techniques are exposed in the Scikit-Learn API, and it's worth sort of exploring both the documentation in Scikit-Learn as well as the standards that are there in the fields of neuroimaging or electrophysiology to figure out the right way that you should go about leaving out subsets of data and validating it on other subsets of data. But moving all of that aside, let's just assume that we've decided that this is, in some sense, a good model, that 66% accuracy is reasonable enough that we think that there is some information in the brain that delineates, I think it was cats and faces. So how do we now figure out why that is the case? How do we figure out what kind of structure this model has found in the data that we've given to it? So what we could do is just look at the coefficients themselves, but these are going to be a little bit imperceptible, right? They're just like a bunch of numbers in a single vector, and that's because we've used this Nifty Masker, which kind of removed all the information about where each of those voxels was in the brain. So here you can see that this coefficients is just, we have a single vector of coefficients, one for each voxel that we fit. So this is where we want to use our Nifty Masker object to transform that vector of coefficients back into the brain's space, back into XYZ space. So to do that, we'll just call inverse transform on the coefficients of the support vector classifier. We'll get back a nifty image like we did before. We'll see that it has the shape x by y by z, so number of voxels in x by number of voxels in y by number of voxels in z by 1. So we just have a single statistical map that we've gotten from this model. And this is an example of how you can save this to a file. This isn't working on the cluster right now because reasons that are not my fault. But what we can do is plot statistical maps of this support vector classifier. So in this tutorial, what we've done is we started off with a bunch of volumetric data that was just voxel activity over time. We took a mask of regions that we cared about. We used that mask to extract the voxels corresponding to regions of interest and then vectorize it so that it was of shape n samples by n features. Then we paired that with a vector of labels saying for each one of our trials what kind of an image was presented. We fed that into a support vector machine that was just a linear classifier. We determined that model was in a quote-unquote good model and sort of worth inspecting into further. And because we decided it was a good model, we then projected the coefficients of that model back into the space of the brain. And we're visualizing that here. And yeah?
If you look at the size of the fMRI mask image or the SSC coefficients, it is much less than the 40 times 64 and 64. So nilearn realizes that it has to put it into that bigger shape. These are EDCs.
Yeah, so that's the masker object tries to be smart about like knowing what space it needs to project data back into, so that it's like an unnecessarily complex thing to do that like we could do that just individually just to keep track of it, but it's
a good thing because
yeah.
you have to manually do it.
Right. Yeah. So and you'll see in a lot of papers they'll visualize these sort of coefficient maps and try to make statements about which regions of the brain are involved in the ability to discriminate between one class versus another. I'm not going to get too like prescriptive about that because that's another big open topic. But this is really meant to just give you an idea of sort of from start to finish what does the neuroimaging machine learning stack look like in the context of nilearn. So then the thing that I want to end with in the last 30 minutes is effectively the same thing, but now moving in the opposite direction and performing a slightly different kind of task. So the first, in the last notebook, we looked at solving the problem of decoding. We went from brain activity and we tried to predict things that happened in the world that corresponded with that brain activity. Moreover, the things that we were trying to predict were categorical variables. We were just trying to predict the class of the image that the person happened to see. For this notebook, we're going to focus on encoding, and we're going to solve a regression problem. So instead of decoding, where we're predicting a set class, we're going to try and predict a continuously varying value. And that's the case because when we're doing an encoding model, the output of the model is a pattern of voxel activity. And that's something that can just continuously vary over time. So this will hopefully give an idea for the considerations that you need to take in order to do this different kind of analysis. The data set that we're going to use is from a Miyawaki paper, and what they did was they showed fMRI participants a series of checkerboard images. So they're just sitting in the machine, they're showing them checkerboard images, they record the voxel activity in response to those images, and then they basically asked two questions. One was, can we model a subset of voxels just using the information about the images that was presented? And then secondly, if we can do that, what are the parts of an image that a particular voxel was paying attention to? This is often called receptive field modeling. It's been around for a long time in animal electrophysiology, and in the last maybe 10 or 15 years has started moving into the world of cognitive, both neuroimaging and also electrophysiology. It's super cool. If you guys want to have a less contentious, more fun conversation about that later on, I also love talking about receptive fields because they're great. And if you're curious about using this for electrophysiology, I can also chat with you about that because that was a big part of what I worked on with MNE Python. Okay, so I'm not going to go into too much detail here. The data or the stimulus that people saw were just 10 by 10 binary images. And we can plot that below here as well. So first, we're just going to fetch the data set. It's just fetch _ Miyawaki. If you're on the Jupyter Hub, it should already be there. Otherwise, it'll go through the downloading process. We can look through the information about sort of how the experiment was set up, the kinds of data that was collected, so on and so forth. This data set is split into a few just sort of random control trials, and then a few where they actually showed the stimulus itself. So we're going to subset the collection of trials that corresponded to actual stimuli. And now we're going to start constructing the matrices that we need to use in order to fit these models with Scikit-Learn. So I mentioned before that we had this Nifty Masker object which can go from volumetric space into samples by features space. There's another object called multi-Nifty Masker that unsurprisingly just does the same thing with multiple Nifty files at once. So here we have a bunch of different files corresponding to different runs, and these just exist in a list in Python. And we can use a multi-Nifty Masker with the mask that's been provided with this data set to operate across that entire list at once. So that's what we're doing right here. We're saying loop through each one of the files that's in this and extract the voxels that we care about, and then vectorize those and return the output as a scikit-learn style object. This is really useful because a lot of times you have voxel data that are in different kinds of files, but ultimately you want to concatenate them. But you only want to do that in memory, like for the purposes of actually fitting the model that you want. Usually that you want to do this because combining all of the 3D data into one file would be extremely large. So you want to apply that mask first. Extract a subset of voxels you care about so that it will fit into memory and you can concatenate it all as a scikit-learn object. So now we'll see that the output of this is itself a list. It's of length 20, corresponding to all of the different runs that were here. And each one is a scikit-learn style object. So if I look inside, so if I look inside, it's now a two-dimensional matrix. So I've got my brain data. The second thing that I now need to do is get my information about the world. And for that, they've given a new set of metadata that describes the kinds of stimuli that were there. These are just CSV files. And if we look inside them, we'll see that these are 144 presentations of a 10 by 10 image. And that 10 by 10 image unraveled corresponds to each column of this stimulus data set. So each column is a pixel, effectively. And just to be able to visualize this, because it's always a good idea to visualize a data set in sort of as raw of a form as possible, we're just going to loop through a few rows of this data set, read in the pixel data, and then reshape it so that it's 10 by 10. And when we plot that, we see this, right? Okay, so this is what the subjects were actually looking at inside of the fMRI machine. And so the question that we're asking is, can we use this information that's right here, this pixel data, in order to predict patterns of activity that we're recording in the brain? So as we saw up there, this data is in 10 by 10 format, but actually the original format of our data is really what we want. It's already of the shape n samples by n features. In this case, features corresponding to individual pixels. So this is something that's quirky to this data set. We're just removing a couple of samples at the beginning and end because there's a slight offset to them. So you can kind of ignore that bit. And then we've vertically stacked all of them here. So now we've taken each individual run and we've combined it all into one big input, well, in this case, output matrix. So this is of shape n samples by n voxels. And now we're going to do the same thing with our stimuli here too. N samples by n pixels. So in the previous example of this, we used something like a Nifty Masker to go from volumetric data into scikit-learn style objects. We're sort of doing something similar here with the image data now, because this is a more complex kind of stimulus representation. We've gone from like a 10 by 10 image to this vector of length 100. And what a lot of people do in their own workflows is that they'll implement their own kind of version of like a Nifty Masker so that they can retain information about like where each pixel goes relative to one another after they've unraveled it into a length 100 vector. So it's something to sort of think about. You can kind of build these sorts of tools that just help you reduce the cognitive load in terms of trying to remember what's actually going on in the data that you have. And if anybody is curious to sort of go through like an exercise of what you might do in order to create such an object for yourself, maybe we can do that as a breakout session in one of the evenings. But either way, right now we've got our data in a scikit-learn friendly shape. We have brain activity in samples by features, and we have stimulus activity also in samples by features. Now what we're going to do is import a different model from the ones that we used before. We're going to use a type of linear regression called ridge regression, which is effectively just a regularized version of linear regression I'm not going to go into tons of details about exactly what that means, but I'm happy to chat about that later on if you're curious. And in this case, we're also going to implement a cross-validation routine as well. So rather than just manually leaving out 50% of our data in one case and 50% as a testing set. Yeah? Slower? Yeah, okay, cool. Tell me if I'm talking too fast in general. I just had coffee, so. Yeah. Do you have a question?
Yeah, I just noticed, so all the models, we're pulling them directly from Scikit-Learn. So nilearn isn't like a re-implementation of these models. So what I'm just not super clear. What exactly, is it just really for organizing the data to put into Scikit-Learn, or what's the purpose?
Yeah, I would say that, I mean, a good principle in general in the open source world is don't reinvent the wheel. Like, try to use tools that are out there if at all possible. And so, I mean, there's also an extra bit of information, which is that like the developers of nilearn are also the developers of Scikit-Learn. So they have, in some ways, like direct access to knowledge of the source to know what's the right way to do this. But I think that their goal is, as you say, not to create a whole new set of machine learning tools that are out there, but to instead say, like, hey, there's this really awesome machine learning toolbox in Scikit-Learn. Let's just figure out a way to allow you to fluidly utilize those tools with neuroimaging data, rather than have to like recreate, our own support vector machine code and our own regression code and everything like that. So I am only kind of scratching the surface of the nilearn API. There's a lot of other stuff you can do in there. And there are some analysis things that are more like specific to neuroscience. But the way that I think about nilearn is a bit of glue that sort of makes your life easier, rather than like its own sandboxed, like fully featured machine learning toolbox. And there are other machine learning toolboxes in the Python ecosystem, I should say, that do have their own kind of like model fitting and do that directly. I think there's one called PyMVPA that does a more kind of like Searchlight MVPA style analysis, and you can kind of pick the one that makes the most sense to you from an API standpoint and from an algorithm standpoint. Does that make sense? Yeah, cool. Okay, so we've imported our ridge regression model here. We're also going to import this k-fold cross-validation object, and this is going to let us take out different subsets of training and test samples on each iteration of the cross-validator. And we're going to use a metric called R² to gauge whether or not our model is making good predictions about the brain activity. So first we're going to instantiate this. We're going to arbitrarily choose 100 as our alpha parameter. This is the regularization parameter for the regression model. And effectively, higher numbers of alpha mean that you are suppressing all of the weights that your model is going to find, all of the coefficients that your model will find. If you set alpha equal to 0, then it will be doing the same thing that linear regression is doing if you just use the vanilla linear regression solution. A lot of times what people will do is use another inner loop of cross-validation to choose the right parameter that they want. That's also something that is quite tricky to do properly, and if anybody's curious about that, we can spend some more time later on talking about that kind of thing. And then so we're going to use 10 splits for our cross-validation object. Just to visualize what this cross-validation is going to do, I'm just showing you each, oh, that's a horrible color. I'm just showing you each iteration of the cross-validation object here on 10 random, on just like a vector of length 10. In blue, I'm showing you the training set for that iteration. And in red, I'm showing you the test set for that iteration. So here you can get an idea for what k-fold cross-validation is doing. It's just taking a different subset that's non-overlapping with all of the subsets and holding that out as a test set each time. If you want to play around with the different cross-validation objects that are there, you could do something like, let's see, where did I import this? Yeah. You can do from scikit-learn.model selection, import, and hit tab, and you'll get access to all of these different cross-validation objects that are out here. So for example, there's I think it's called shuffle split object that just shuffles the data and then splits it into two subsets. So we could do CV equals shuffle split. We'll keep the same number of splits. We'll say the test we'll say the test size equals 0.1. So that means leave out 10% of the data for testing data on each iteration. And now if we create the same plot, we can see now that it's leaving out different subsets of the data and is apparently doing something really wonky with the first two cross-validation iterations. So you can use this to kind of visualize what the behavior of your cross-validator is going to be for different kinds of cross-validators that are out there. I'm going to delete that and redo this with kfold. Okay. So now what we're going to do is we're going to loop through all 10 iterations of our cross-validation object. And we're doing that with this for loop here. So you can just do for train, comma, test in the object.split and then give it some data. And it tries to be clever about knowing like which indices it needs to leave out in order to satisfy whatever requirements you've given for the cross-validation object. And you can kind of ignore TQDM right here. This is just so that we get a progress bar while it happens. On each iteration, we're going to shape our input data so that they're of samples by features. And remember, in this case, the input data are pixel information. The output data are voxel activation. So we're going to split our data into training and test sets here. We're going to fit that ridge regression model using our training data. And we're going to generate predictions of our ridge regression model on the testing data. So one thing to remember is that y_train, this is our output, this is our this is our voxel activity. And this is actually a two-dimensional array. So it's of shape n samples by n voxels. So when we fit this model here and we include a two-dimensional array as output, what it's actually doing is fitting a bunch of different models, one for each column that's there. So y_train in this case is samples by n voxels. So when we do it this way, the model itself is actually going to have a different set of coefficients for each voxel. So we've fit 5,400 models. Each one has its own set of coefficients of length 100, one coefficient for each pixel. And the other thing that we've done is that we've appended the score of that model fit on each of those iterations. So what we'll do is take the mean value of the score across all of the cross-validation loops, and then we'll define some sort of an arbitrary cutoff that just says what scores we want to actually visualize. So now cut score is of length number of voxels. So this is one score per voxel. And it's just the cross-validation score that we've gotten for each of those voxels. 0 just means that it did a really bad job, basically. Now we're going to use that Nifty Masker to invert those scores and project them back into voxel space. And we'll use that with the masker object that we created before. And now we can make a statistical plot of which voxels did well, or how each voxel did in prediction using this ridge regression setup. So here you can see there's kind of a distribution of scores, some of them better, some of them worse. It's common to use thresholding in order to plot subsets of this image. And so we can, so we'll do that in a second. But first I just want to give maybe like an idea of what we mean by a good score or a bad score. So what I'm going to do is for one of the quote-unquote bad voxels, or a voxel that doesn't have a very good score, I'm going to make a scatter plot of, for each trial, the actual value of that voxel activity on the x-axis and the predicted value of that voxel activity on the y-axis that our model actually predicted. And so this is for one voxel. Each point is a different sample or a different presentation of the data. And here we actually get two interesting pieces of information, I think. The first one is that it doesn't look like there's any relationship between two things. And that's what we would expect for a model that does a bad job of generating predictions. If the model's predictions have nothing to do with what's actually happening in the world, it's not a good model. The second thing that we may learn from this is that it looks like there might be a bug somewhere in the code, because we have this line of plots here at the top, or this line of dots here at the top. And I don't know why it's there, but it certainly doesn't look like sort of normal naturalistic kind of data. So we're not going to go in and debug that right now, but I think it's just another reason that it's quite useful to visualize things as much as possible to make sure that things are sort of happening in the way that you would expect them to happen. Now I'm going to generate this same plot for a voxel that has a relatively speaking high score. And here we can see that there is maybe a trend here that shows that there's like a positive relationship between the predicted and the actual value. Whether or not that is driven by the fact that we have this like weird line of prediction scores at the bottom is anybody's guess. My guess is that this line down here makes it seem like we have a higher prediction score than we actually do. This is something that's relatively common in encoding models. So you should always create these kinds of plots. I'm sort of showing it here because like you should always look at your data in this kind of way to make sure that if you're only calculating correlation values or R-squared values, sometimes it may be telling you things that are different from what you think it's telling you. So general rule, like visualize your raw data as much as possible, at least when you're debugging your code. Okay, so now we're just going to set a threshold of 0.2 and visualize the same statistical map that we looked at before. I'm adding a little bit of extra machinery here just so that we can visualize a couple of specific voxels. So here, this is the same statistical map that we just looked at, but now I said anything that has a score less than 0.2, just remove it, so that we can highlight the voxels that are most well predicted. And then we're adding a little colored bar around here because around these 4 voxels, because we're going to treat these as sort of voxels of interest. So if you remember in classification, first we looked at the ability of each classifier to generate those predictions of one class versus another. And then after we had concluded that that classifier did well, we inspected the coefficients of the model to try and figure out which voxels were important in generating that classification. In this case, we have a bunch of different models, right? We fit like 5,000 different models, and we have a score for each model. So what I'm showing you here are the subset of models that did a relatively good job on this regression challenge. And so what we'll do is focus on 4 of the models that did relatively well and inspect the coefficients of those models to say what kind of structure did those models find in the relationship between this pixel information and the neural activity that was here. So this last bit here effectively redoes the model fitting that we did before, just so that we can visualize the kinds of structure that the model is finding. And I'm also giving you a couple of different options for the type of model that you might fit. So there are lots of different kinds of regression models that are out there. The one we used before was ridge regression. There's another popular version of regression called Lasso Regression, also known as L1 regression. It achieves a similar kind of result, except that it leads to coefficients that are more sparse. So you're going to have more zero coefficients. You're going to have fewer coefficients with a large value attached to them. You should choose the one that you think makes the most sense based on your assumptions about the data effectively. In addition, these are sort of helper objects that include a little bit of cross-validation inside of the object itself so that we don't have to do that on our own. Okay. Oh, and then the other thing that I'm doing here is this is just a way that you can try and choose a regularization parameter within one of these objects here. So I'm actually giving this ridge regression object a list of alpha values. And what it will do is loop through each alpha value, fit the model, and predict it on held out data. And it'll do that for every possible value of alpha, and then it will retain the value that gave you the best model performance. So what we'll do is we'll fit these voxels right here. Those just correspond to the indices that we've highlighted up there. And then I'm just going to mark a single pixel on the 10 by 10 grid so that we can have reference between all of the different visualizations that we'll show. So for each voxel, we're going to pull out the fMRI data for that voxel. We're going to fit a model with the pixel data as input and the fMRI data for one voxel as output. Then we're going to reshape the coefficients of that model so that they're in a 10 by 10 grid, which is the shape that the actual stimulus information came in. And then finally, we're going to visualize what that 10 by 10 grid looks like. And we're going to do that for each of the voxels that we've got there. So what I'm showing here is the receptive field for all of the voxels that we've highlighted. In this case, the relative position of these plots corresponds to the relative position that the voxels have to one another. And higher values or more colored values mean that pixels that are in those locations tended to be correlated with increases in the voxel activity for that voxel. Another way that you would describe this is that, for example, on the top left, this blue plot shows the receptive field for that voxel. It's the kind of stimulus that will tend to correlate with an increase in activity in the voxel. A lot of people in the encoding world, when they talk about this, they use very shall we say, like causal language. They say like this is the stimulus that will cause an increase in activity in the voxel. I think that's probably a mistake. At the end of the day, these are just like correlations that we're pulling out of the data. So be careful about like how you word your conclusions when it comes to this kind of stuff. But what this does tell us is, in some sense, like what is the area of visual space that this voxel cares about, or that this voxel tends to have an increase in activity when it's there. Okay, so just to sort of recap everything that we've talked about here, we began by talking about how you can go from neural activity that's stored onto disk and read it into memory in Python so that you can incorporate it into the sort of neuroimaging stack. Then we talked a little bit about how you can connect that neuroimaging stack with the machine learning Scikit-Learn stack in Python using things like a Nifty Masker. We then talked about going through sort of one round trip of analysis in the context of decoding, so trying to understand how you can decode information about the world using patterns of brain activity. And then we visualized which regions of that brain were correlated with differences in one category of image versus another. And then finally, we looked at an encoding model where we used pixel information of 10 by 10 images that were shown to people to ask, can we predict patterns of activity in the fMRI voxels using these pixels. And then we looked at a subset of voxels that were well predicted and then visualized the receptive fields that the models had fit for those voxels. So as I said before, this is very much scratching the surface. The world of machine learning and in neuroimaging and in electrophysiology is vast, and it's getting bigger and bigger every day. Like right now, there are a bunch of folks in New York City talking about all sorts of cool machine learning and neuroscience stuff at the CCN conference. I also didn't talk about at all more complex model fitting methods like nonlinear models. Probably most particular of interest are neural networks. But I think that you did a tutorial about that yesterday. I would say for any of this stuff, if you guys have questions about it, or if you're curious about how to implement these things for your data, or if you want to try out some of these algorithms in some of the hacking sessions, I'm more than happy to chat with you about it, and I'm sure that all of the instructors are too. And I guess if you guys want to chat or have questions, then we can talk about it over lunch. So thanks.
Before we go to lunch, thank you.